Welcome to the Science Experience, your source for the most current and impactful research and preprints in science. Before we embark on today's deep dive, please take a moment to like this video and subscribe to our channel. Your support truly helps the Science Experience continue bringing you these deep dives into cutting-edge science. Today, we're embarking on a journey into the hidden biochemical landscape of your own body to unpack something truly mind-bending. It challenges everything you thought you knew about one of the most feared molecules. Cyanide. Yes, you heard that right. We're talking about cyanide being produced inside your own body, acting as a signaling molecule.
It's quite a revelation, isn't it? Really turns things upside down. Our mission for this deep dive is to unravel how something conventionally viewed as, a deadly poison can actually be generated endogenously, right inside mammalian cells and tissues. We'll explore the intricate mechanisms behind its production, its surprising physiological roles, and crucially, what happens when this delicate balance goes awry. All of this fascinating insight comes from a recent publication in Nature Metabolism, and it's really turning traditional toxicology on its head.
Yeah, when most of us hear cyanide, our minds immediately jump to poison, danger, maybe a dramatic movie scene. It's the ultimate toxic villain, sort of, in popular culture. But this incredible new research doesn't just challenge that idea, it fundamentally redefines it.
Indeed. And to truly grasp the significance, we need to talk about a class of molecules called gasotransmitters. These are small gaseous molecules like nitric oxide, NO, carbon monoxide, CO, and hydrogen sulfide, H2S. Our bodies naturally produce them, and they aren't just waste products, they act as vital signaling molecules regulating, everything from blood pressure to brain function. What the study proposes, and I think convincingly demonstrates, is that cyanide, which we've historically seen solely as a cytotoxic external agent in mammals, it should now be considered part of this elite group of endogenous regulatory gasotransmitters.
Okay, let's unpack this. If cyanide is an internal messenger, where exactly is this surprising molecule found in the body? Is it widespread or confined to a specific area?
Oh, it's far from isolated. This research reveals that cyanide has been detected endogenously across a really wide range of mammalian tissues. We're talking about your liver, spleen, kidney, lungs, even your brain. It's also found circulating in the blood. Beyond tissues, it's present in a diverse array of human cells too, like hepatocytes, those are liver cells, lung and colonic epithelial cells, and even immune cells, peripheral blood, mononuclear cells, neutrophils. It's quite broad. What's particularly notable, though, is that liver tissues and cells consistently show the highest rates of cyanide production. So the liver seems to be a major hub.
Okay, so it's pretty active throughout the system, which of course raises the important question. If it's being made inside us, how exactly does our body manage this, and where does this surprising cellular factory operate?
Right, good question. The study pinpoints a critical starting material, glycine. This is a very common amino acid, one of the building blocks of proteins. An experiment showed that simply adding glycine significantly stimulates cyanide generation in cells and tissues. What's really remarkable is that no other common proteinogenic amino acids had this effect, just glycine.
So it's specific to glycine.
Exactly, highlighting its unique role. And this isn't just some random chemical reaction happening, it's enzymatic. Specific proteins in our bodies are actively involved in manufacturing it. And when researchers looked closely inside the cells, using techniques like confocal microscopy, they found the primary production site, lysosomes.
Lysosomes. Aren't those usually thought of as the cell's recycling centers, breaking down waste?
That's the common view, yes. But rigorous experiments isolating lysosomes and comparing them to the cell's main fluid confirmed that cyanide production, especially when stimulated by glycine, happens right there. And what's more, the integrity of these lysosomes is absolutely essential. If you disrupt them, cyanide production just drops off.
So the cell's cleanup crew is also secretly making cyanide. That's quite a twist. Here's where it gets really interesting.
It really is fascinating. And the conditions needed are quite specific too. It requires the low acidic pH that's characteristic of lysosomes, typically around pH 4.5. And it also relies on what's known as peroxidase activity. Peroxidases are enzymes that help certain chemical reactions along. Specifically two enzymes, myeloperoxidase, MPO, and peroxidasin, PXDN, which are known to be preferentially located in lysosomes, appear to be the key players here. Basically, these peroxidases use hydrogen peroxide and chloride as substrates to produce hypochlorous acid, HOCl. This HOCl then reacts with glycine in that acidic lysosomal environment to ultimately yield hydrogen cyanide, HEN, and carbon dioxide.
And then this hydrogen cyanide it can just leave the lysosome.
Yes, exactly. Because it's a gas, HCN, it can easily diffuse out of the lysosome into the cytosol, the main cell fluid, and potentially other cellular compartments too. It might even diffuse right out of the cell to act as a paracrine mediator, signaling to neighboring cells. It's potentially a very mobile messenger.
Okay, knowing it's produced internally and it can move around, what does this all mean for our bodies? What roles does this newly discovered internal cyanide actually play?
Well, first off, like any potent substance, our bodies have a system to keep things in check. There's a main detoxification enzyme for cyanide called thiosulfate sulfur transferase, or TST. Some people call it rhodonase. Its main job is basically to maintain that delicate balance, preventing cyanide from accumulating to toxic levels, keeping endogenous levels where they should be.
Right, a built-in safety mechanism.
Precisely. Now, for its functions, one of the most fascinating ways endogenous cyanide seems to work is by modifying proteins. It adds a cyanide group, a CN group, to the sulfur atom and specific amino acids called cysteine residues within proteins. This modification is called S-cyanylation, and this isn't just theoretical. It's detectable even at baseline levels in cells and in mice. And it significantly increases when you add more glycine, boosting cyanide production. So this suggests cyanide isn't just floating around passively, it's actively remodeling the intracellular landscape of protein modifications. It's changing how proteins behave.
That's quite intricate. Could you maybe offer an analogy? Like, how does this S-cyanylation work?
Yeah, absolutely. A good way to think about S-cyanylation is like a redox switch on a protein. Depending on the specific protein it modifies, this switch can either turn the protein's function on, activating it, or turn it off, inhibiting it. For instance, the study showed it actually increased the activity of one key enzyme involved in energy metabolism, GAPDH, but it inhibited another enzyme, GPDH, which is involved in fat metabolism. So it's not a simple on-off for everything, it's targeted.
So it's a crucial aspect of its regulatory role, fine-tuning different cellular processes.
Exactly. And beyond just tweaking individual proteins, these low endogenous concentrations of cyanide seem to have broader stimulatory effects. When glycine supplementation increases cyanide levels slightly, it significantly enhances mitochondrial bioenergetics. That means better energy production by the cell's powerhouses. Things like electron transport and ATP generation get a boost. It also seems to promote cell proliferation or cell growth.
And the opposite is true, too, if you lower it.
Correct. Conversely, if you reduce endogenous cyanide levels, either by using molecules that kind of mop it up, scavengers, or by inhibiting its production pathway, these vital functions like energy production and cell growth are impaired.
This is a really important takeaway, then. It sounds like it's all about the concentration, the amount.
Precisely. That's perhaps the most critical point. Just like with the other gasotransmitters we mentioned, NO, CO, HUS cyanide's beneficial effects are highly concentration dependent. There's an optimal physiological range, a kind of sweet spot. If there's too little, maybe because that detoxification enzyme, TST, is working overtime, or if there's too much, if it starts to accumulate beyond that sweet spot, then it can impair cellular energy production and proliferation. It flips from beneficial to detrimental.
It's a classic example of the dose makes the poison, but applied internally.
Exactly. It's the fundamental insight here. It completely reframes how we should view cyanide, not just as a poison, but as a molecule that in tiny controlled amounts is vital. And there's more. Its protective effects are quite remarkable too. Endogenously generated cyanide also exerts significant cytoprotective effects. Protection for cells, glycine supplementation, which boosts internal cyanide, and even carefully controlled low concentrations of external cyanide or related cyanogenic compounds. Things like mandolonitrile, linamarin, or amygdalin, which you might know from apricot kernels. They were shown to protect liver cells from cell death, specifically death induced by hypoxia, low oxygen, and then reoxygenation. This mimics the kind of stress cells experienced during, say, a heart attack or a stroke.
Wow, so it can protect against that kind of damage. Is there data from actual animals, not just cells?
Yes, and that's crucial. The study has compelling in vivo data from living organisms, to back this up. First, they confirmed detectable cyanide concentrations in normal mouse blood. The levels, around 585 nanomolar in males, 364 in females, are actually quite comparable to what's found in healthy non-smoking humans, which is about 540 nanomolar, so it's normally there. Then, they administered glycine or amygdalin to these mice. This increased their blood cyanide concentrations, but only up to around 1 micromolar, still in the low non-toxic physiological range. And here's the key part. In mouse models of myocardial ischemia-reperfusion, basically mimicking a heart attack, and also hemorrhagic shock. Giving these mice low-dose amygdalin significantly reduced organ damage. It showed a direct protective effect in living animals.
That's huge. And did the dose matter there too?
Absolutely.
Hmm.
Importantly, that bell-shaped dose response was also observed in these protective effects. Too little amygdalin didn't help much, too much could potentially be harmful, but the right low dose was protective. It reinforces that optimal concentration is key, both for basic function and for protection.
Okay, so this delicate balance is clearly vital for health. But what are the consequences when the body gets it wrong? When the bell tips too far to the high side? Are there real-world examples?
Yes, absolutely. A tragic but very illustrative example is a condition called non-ketotic hyperglycemia, or NKH. This is a devastating, rare genetic disease. It's characterized by a pathological buildup of glycine in the body way above normal levels because of mutations in the enzymes that usually break glycine down.
And then glycine is the precursor for cyanide production.
Exactly. You see the connection. The study investigated this link and found something dramatic. They took fibroblasts, skin cells, from individuals with NKH and compared them to cells from healthy controls. The NKH cells showed markedly elevated endogenous cyanide levels. We're talking an astonishing 30-fold higher production rate compared to the control cells. So in these NKH cells, because of the massive glycine overload, cyanide clearly reaches cytotoxic or cell-damaging levels.
So the very mechanism that's beneficial at low levels becomes damaging at high levels in this disease. It causes mitochondrial dysfunction, reduced energy, impaired cell viability, exactly what you'd expect from cyanide poisoning, but generated internally.
Precisely. And what's truly insightful and offers maybe a glimmer of hope is that the study showed that by reducing these high cyanide levels in the NKH cells, for example, by using a compound called hydroxychloroquine, which makes the lysosomes less acidic and thus inhibits cyanide production, the cellular function, things like energy production and viability, actually improved.
Wow. So it wasn't just a correlation. They showed reducing the excess cyanide helped the cells.
Correct. It strongly suggests the cyanide overload is a key part of the problem in NKH. This pathological condition starkly illustrates the upper limit of cyanide's beneficial range. It powerfully underscores why maintaining those optimal levels is absolutely crucial for health. It's a real-world validation of that bell-shaped curve concept in a disease context.
So let's pull back a bit. What does this all mean for our fundamental understanding of biology? This research seems to fundamentally shift our perspective. It moves cyanide from just being a poison in a textbook to placing it alongside nitric oxide, COH2S, as a critical mammalian regulatory gasotransmitter. That's a huge reevaluation.
It truly is. It's a paradigm shift, really.
Yeah.
And it also has these fascinating evolutionary implications. Think about it. Cyanide and hydrogen sulfide, H2S, existed on Earth billions of years ago, long before there was much oxygen in the atmosphere. This suggests that enzymatic systems capable of producing these gases, albeit at low, controlled rates, evolved very early on. They likely have ancient, conserved roles in biological function. We know about them in bacteria and plants, and now we see these incredibly old molecules are still playing vital roles, despite their potential toxicity, right inside us mammals.
This deep dive really just reveals the incredible precision, the complexity, maybe even the unexpected resourcefulness of our own biology. It raises so many important questions for future exploration, doesn't it? Like, how is this endogenous cyanide production precisely regulated from moment to moment? Are there specific target proteins that get acylated only under certain conditions, like during stress or exercise, perhaps? And how do these interactions influence our overall metabolism and physiology in different conditions throughout our lives? There's so much more to learn.
Absolutely. And beyond the fundamental science, this work potentially opens up new avenues for therapeutic strategies, especially for conditions involving cellular stress and injury, like heart attack, stroke, maybe even shock, where boosting these protective mechanisms carefully could be beneficial. And as we saw, it offers completely new insights into the pathophysiology of devastating diseases like NKH, which might lead to novel treatment approaches.
Ultimately, what stands out to me is the thought, maybe our bodies are still holding other biochemical secrets. Or are they employing other molecules we currently just think of as toxic for surprising beneficial purposes? What else might be hidden in plain sight, just waiting for us to uncover its secrets? Makes you wonder. Remember to like this video and subscribe. Your support truly helps the Sand Science Experience continue these in-depth explorations of cutting-edge science. Thank you for joining us on this deep dive into endogenous cyanide. We hope you gained some valuable insights and definitely some surprising facts today. This has been the Sand Science Experience.